Ever wondered how we can see inside the human brain without surgery? This is where the fascinating field of neuroimaging comes into play. It's like having a window into the brain, giving us the ability to observe its structure, function, and connectivity in an entirely non-invasive way. This is crucial in both research and clinical contexts, helping us understand the brain's intricate workings and diagnose a range of neurological conditions. There are several types of neuroimaging techniques, each with their unique strengths and applications. From magnetic resonance imaging, or MRI, which gives us detailed images of the brain's structure, to functional MRI and positron emission tomography that measure brain activity, to diffusion tensor imaging that maps out the brain's connectivity. Each technique is a powerful tool in its own right, and together they provide a comprehensive view of the brain. So let's embark on this journey to uncover the mysteries of the brain through the lens of neuroimaging. Imagine you could take a detailed snapshot of the brain's structure. That's exactly what MRI and CT scans do. These two techniques are cornerstones of structural neuroimaging, revealing the intricate architectural landscape of our minds. Magnetic resonance imaging, or MRI, uses a powerful magnetic field and radio waves to generate detailed three-dimensional images of the brain. This allows us to visualize both gray matter, where the brain's neuronal cell bodies reside, and white matter, the highways of nerve fibers that connect these cells. This remarkable technology can reveal subtle changes in brain structure that may be linked to various neurological conditions. Computed tomography, or CT, on the other hand, uses a series of X-ray images taken from different angles around the body to create cross-sectional images of the brain. While not as detailed as an MRI, kinetic scans are quicker and can provide valuable information, especially in emergency situations. Let's consider a case study to illustrate the power of these techniques. Imagine a patient who presents with chronic headaches and visual disturbances. An MRI scan reveals a tumor pressing on the optic nerve. This diagnosis would have been impossible without the detailed images provided by the MRI. The patient then undergoes surgery, and the symptoms resolve post-operatively. This real-world example underscores the critical role of MRI and CT scans in diagnosing structural abnormalities in the brain. These technologies are not just limited to identifying problems, they also provide a foundation for understanding the normal brain. By studying healthy brains, we can establish a baseline to identify deviations linked to neurological conditions. This comparative approach helps us to understand the root causes of these disorders and drives the development of new treatments. MRI and CT scans, thus, serve as critical tools for diagnosing structural abnormalities in the brain. They offer a window into the brain's intricate structure, providing us with invaluable insights into its inner workings. Through these techniques, we continue to deepen our understanding of the complex and fascinating world of neuroscience. But what if we want to see the brain in action? Enter functional neuroimaging. Functional magnetic resonance imaging, or fMRI, is a game-changer in the world of neuroimaging. It uses the same principles as an MRI, but rather than capturing static images, it measures changes in blood flow to different parts of the brain. This blood flow is linked to neural activity, so when a brain region is in use, it lights up on the fMRI. Consider this. Researchers conducted an fMRI study on individuals with social anxiety disorder. Participants were asked to perform a public speaking task, a situation that tends to trigger anxiety. The fMRI scans revealed increased activation in brain regions associated with fear and anxiety in individuals with social anxiety disorder compared to those without, providing insight into the neural mechanisms underlying the disorder. Now let's talk about positron emission tomography, or PET. A PET scan involves the injection of a small amount of radioactive tracer into the bloodstream. This tracer travels to the brain and emits signals that are picked up by the PET scanner. The signals are then converted into images that show how different brain regions are functioning. For instance, a patient with suspected Alzheimer's disease underwent a PET scan with a radiotracer that binds to amyloid plaques, the hallmark of Alzheimer's in the brain. The PET imaging confirmed the presence of significant amyloid deposition in the brain, supporting the diagnosis of Alzheimer's disease. Finally, we have electroencephalography or EEG. This technique measures the electrical activity of the brain using electrodes placed on the scalp. These electrodes detect the tiny electrical charges that result from the activity of the brain cells. EEG is particularly useful in diagnosing and monitoring conditions like epilepsy, sleep disorders, and other neurological problems. Functional neuroimaging techniques, therefore, provide a dynamic picture of the brain, revealing how different regions interact and respond to various tasks. For a more specialized view, we turn to techniques like diffusion tensor imaging, or DTI, and magnetoencephalography, known as MEG. DTI is a type of MRI that delves into the brain's white matter tracts, the brain's internal wiring that facilitates communication between different regions. By tracking the diffusion of water molecules, DTI can map out these tracts, giving us a detailed view of the brain's structural connectivity. This is particularly useful in understanding conditions where this connectivity may be impaired, such as in traumatic brain injuries or neurodegenerative diseases. Let's consider a case study. A patient presents with motor deficits following a traumatic brain injury. Traditional imaging techniques may not reveal the full impact of the injury on the brain's structure. However, with DTI, we can assess the integrity of the white matter tracts. In this case, DTI showed disruption of tracts in regions associated with motor function, correlating with the patient's clinical symptoms. This information is invaluable in guiding rehabilitation efforts and monitoring recovery. Now let's shift gears to MEG. MEG is a non-invasive technique that measures the magnetic fields generated by neuronal activity. It offers an exceptional temporal resolution, allowing us to study the brain's dynamics in real time. This makes MEG particularly valuable for understanding conditions like epilepsy, where timing is crucial. By tracking the millisecond by millisecond activity of the brain, MEG can help pinpoint the exact onset and spread of a seizure within the brain. Together, DTI and MEG provide a complementary view of the brain. DTI enlightens us on the brain's structural connectivity, and MEG provides a window into its temporal dynamics. They are powerful tools in our neuroimaging arsenal, helping us unravel the intricate workings of the brain. DTI and MEG offer unique insights into the brain's structural connectivity and temporal dynamics. Sometimes one lens isn't enough. That's where combined approaches come in. In our journey through the landscape of neuroimaging, we've encountered an array of techniques, each with its own strengths and limitations. But what if we could harness the power of multiple techniques simultaneously? Enter multimodal neuroimaging, a combined approach that integrates several types of data, offering a more comprehensive perspective on the brain. Multimodal neuroimaging brings together the best of structural, functional, and connectivity data, transcending the limitations of individual techniques. By combining methods, we can correlate structural changes with functional alterations and relate these to the brain's connectivity patterns. This powerful synergy allows us to explore the intricate structure-function relationships in the brain with unprecedented detail. Consider a situation where a neuroscientist is studying a neurodegenerative disorder such as Alzheimer's disease. Structural MRI can reveal atrophy in specific brain regions, while PET scans can identify the buildup of harmful amyloid plaques. Meanwhile, fMRI can show altered brain activity patterns, and DTI can expose disruptions in white matter tracts. By integrating these diverse data, researchers can gain a holistic understanding of the disease's impact on the brain, from macroscopic structural changes to microscopic alterations in brain activity and connectivity. Similarly, in the field of psychiatry, multimodal neuroimaging can illuminate the underpinnings of mental disorders. For instance, in individuals with depression, structural MRI may show reduced hippocampal volume, while fMRI could reveal altered activity in mood-regulating brain circuits. By correlating these findings with connectivity data from DTI or MEG, researchers can form a comprehensive picture of the disorder's neural basis. The beauty of combined approaches lies in their versatility. Whether it's studying normal brain function, investigating neurological disorders, or informing treatment strategies, multimodal neuroimaging opens a window into the brain's complex landscape. In conclusion, the future of neuroimaging lies not in choosing one technique over another, but in integrating them to form a richer, more nuanced understanding of the brain's workings. By integrating different types of data, we can form a more complete picture of the brain. So we've taken a brief tour of the brain through the lens of neuroimaging, from the intricate anatomical details revealed by MRI and CT to the dynamic neural activities captured by fMRI, PT, and EEG. These techniques offer a fascinating glimpse into our brain's inner workings. We've also explored how DTI maps white matter tracks and how MEG provides high-resolution insights into brain dynamics. With combined approaches, we can integrate structural, functional, and connectivity data for a more comprehensive understanding of the brain. Looking ahead, the future of neuroimaging holds great promise. Advances in technology and computational methods will likely lead to even higher resolution imaging, more accurate mappings of brain connectivity, and innovative ways to integrate and analyze multimodal data. This will not only deepen our understanding of the brain, but also have profound implications for diagnosing and treating neurological and psychiatric disorders. As technology advances, we can only expect to see more exciting developments in neuroimaging, bringing us closer to unraveling the mysteries of the brain. So that's it for today. Join us in the next episode as we explore the ethical considerations in neuroscience research and practice, navigating the complex intersection of science, ethics, and society. Don't forget to like, share, and subscribe, and comment your thoughts below. Until next time, keep learning.